In this class, I'm explain about Human Genome Project. In this topic, we discuss about meaning of Human Genome Project, history of Human Genome Project, aims of Human Genome Project, goals of Human Genome Project, and human genome size and important feature of human genome and Human Genome Project mapping and Human Genome Project sequencing. First one, meaning of Human Genome Project. The Human Genome Project is an international collaborative research program which started in 1990 and completed in 2003. The term genome was introduced by H. Winter in 1920. It refers to one complete copy of the genetic information, that is DNA, or one complete set of chromosome, that is monoploid or haploid of an organism. The term genome, it refers to one complete copy of the genetic information or one complete set of chromosome of an organism. And the term genomics denotes mapping, sequencing, and functional analysis of genome. The genomics term coined by T.H. Rodericks in 1987, and the Human Genome Project was launched on 1st October 1990 for sequencing entire genome of 2.75 billion nucleotide pairs. And this mega project was a 13-year project coordinated by the US Department of Energy and the National Institute of Health during the early years of the Human Genome Project. The Wellcome Trust in UK, it becomes a major partner. Additional contributions come from Japan, France, Germany, and China and other. And the project was complete in 2003. James Watson was the first director of HGP, and after a period of 2 years, he was replaced by Francis Collins in 1993. Two important scientists associated with the Human Genome Project were Francis Collins, director of Human Genome Project, and J. Craig Venter, founding president of Celera Genomics. And Human Genome Project was closely associated with rapid development of a new era in biology called as bioinformatics. And next one is the history of Human Genome Project. In US, the Human Genome Project was carried out by the Department of Energy, that is Human Genome Program directed by Ari Patrinos, and National Institute of Health Human Genome Research Institute directed by Francis Collins in 2001. Craig Venter, CEO of Celera Genomics, co-announced the completion of sequencing of the human genome. And next one is the aims of Human Genome Project. The project was aimed for the benefits of humankind. Generation of biologists and researchers have been provided with detailed DNA information that will be key to understand the structure, organization, and function of DNA in chromosomes. And next one is goals of Human Genome Project. Goals. Goals of Human Genome Project. Human Genome Project had many goals. Some of the important goals are to identify all the approximately 20,000 to 25,000 genes in human DNA, and to determine the sequence of the 3 billion base pairs that makes up human DNA, and to store his information in database, and to develop improvised tools for data analysis. And next goal is to transfer related technologies to other sectors such as industries. And sixth one, to address the ethical, legal, and social issues that may arise from the project. Next one is the important feature of Human Genome Project. The human genome contains 3164.7 billion nucleotide bases. And second one is the average gene consists of 3000 bases, but gene size vary greatly. The largest human gene is dystrophin containing 2.4 million bases. Third one, the total number of genes in the genome is estimated at 30,000, and all nucleotide bases are exactly the same in all people. And fourth one, functions of about 50% of the discovered genes are still unknown. And fifth one is less than 2% of the genes of the genome codes for proteins. And sixth one is chromosome 1 has most genes and the Y has the fewest. The chromosome 1 has most genes, number is 2968, and the Y chromosome is 231. And seventh important feature is repeated sequences 80, 80, 80 or GC, GC, GC make up very large portion of the human genome. And last one is scientists have identified about 1.4 million locations where single base DNA differences occurs in human. And next one is human genome size. The human genome is an organism's complete set of deoxyribonucleic acid, that is DNA, a chemical compound that contains the genetic instructions needed to develop and direct the activities of an organism. The human genome contains approximately 3 billion base pairs which resides in 23 pairs of chromosomes. And next one is the Human Genome Project mapping. Before beginning a sequencing project of the human genome, it was first necessary to produce a good framework map. Two general methods were developed for mapping human genome. First one, standard method, and second one, whole genome shotgun method. First one, the standard method involves finding a segment of the genome and locating where it belongs. Genetic maps based on the combination frequencies between markers are useful in ordering genes. And molecular markers like RELP means restriction fragment length polymorphism, and STS, sequence tagged sites, and SNPS, single nucleotide polymorphism, has been used in the mapping human genome. And the whole genome shotgun sequencing method involves the sharing of genomic DNA followed by cloning to produce a genomic library. This is followed by the sequencing of cloned DNA fragments at random, followed by shotgun assembly, that is the assembly of the fragment sequences into larger units on the basis of their overlaps. Groups of cloned DNA segments that can be aligned in an overlapping fashion to cover a region of the human genome are referred as the contigs. Contig refers to the set of overlapping DNA segments derived from a single source of genetic material from which the complete sequence may be deduced. And next one is yeast artificial chromosome. YACs were initially used as cloning agents where primary task was mapping. However, as the emphasis of the project shifted to sequencing, and bacterial artificial chromosomes were used. And next last topic is Human Genome Project sequences. The sequencing means determining the exact order of the base pairs in a segment of DNA. And the primary method used to Human Genome Project to produce the finished versions of the human genetic code is map based on BAC, that is bacterial artificial clone based sequencing. The human DNA is fragmented into pieces that are relatively large, cloned in the bacteria and stored for replication as required. And a collection of BAC, that is bacterial artificial genome clones containing the entire human genome, is called the BAC, bacterial artificial clone library. In this method, each bacterial artificial clone mapped to determine the location of that fragment in human chromosome, and then the DNA letters are sequenced from each clones and their spatial relation to sequenced human DNA in other back clones. For sequencing each bacterial artificial chromosome, clone is cut into still smaller fragments that are about 2000 base in length. These pieces are called subclones. A sequencing reaction is carried out on these subclones with the help of a computer. Then the short sequences are assembled into cortex stretches of sequence of the clones. That's about the Human Genome Project. Thank you.